Good morning to all. I am Dr. M. Mariraj, Assistant Professor, Department of Botany, Sri Kaliseri College, Sivasi. Today I am going to talk about the general characters of bacteria. Bacteria is a prokaryotic organism. The bacteria sometimes is the pathogenic bacteria, sometimes non-pathogenic bacteria. Bacteria is present everywhere of the earth. Bacteria is the first living organism is evolved in the earth. The bacterial introductions. Bacteria are unicellular microorganisms ranging in the length a few micrometers to half millimeters. Bacteria can be found in almost every ecosystem on the earth. The bacteria are found 2 billion years and now is 3.0 billion years ago. Some bacteria are pathogenic, cause the disease for the plants and humans and animals. Bacterial history. The bacteria was first observed by the Anton Leeuwenhoek in 1676. The term of bacteria is Greek for small stuff was first used in the 1938. The bacterial history. The Robert Koch and Louis Foster were first to discover that bacteria cause the many diseases in the mid-19th century. The first antibiotic used to treat the bacterial disease was made by the Fall Eckridge in 1910. It was used to treat the syphilis. The evaluation of bacteria. The bacteria is thought to be first forms of life about 4 billion years ago. It is believed that the both domain of archaebacteria and domain of eukarya evolved from bacteria. This is the classification of bacteria. This is the first ancestor prokaryote is found. That is the bacteria is a prokaryote. This is the only nuclear region is present in the bacteria. Then ancestral eukaryote will form. This is the eukaryote. This is the nucleolus and is present. The multicellular organisms. Then chloroplast was formed to evaluate the plants. Then after mitochondria organelle formed to producing the protozoa, fungi, animal and multicellular organism. Then another different prokaryote is formed by archaebacteria. This archaebacteria is different from bacteria because of the cell wall content. So archaebacteria have the peptidoglycan is the important layer. So archaebacteria doesn't have the peptidoglycan layers. Archaebacteria, some nucleic genetic characters also differ from the bacteria. Morphology. Bacteria cells are prokaryote lacking the nucleus and only nuclear region is present. The complex organelles is present. The bacteria have the flagella and pili structures. Bacteria has the ribosomes and pili structure, flagella, DNA and plasmidium also present. The plasma membrane is present in the outside the cell wall. They have the cell membrane and cell wall made up of the peptidoglycan. This is the gram positive cell walls. Another one is gram negative cell wall. Gram positive cell wall is the peptidoglycan layer is very thick and gram negative cell wall is the only thin amount of peptidoglycan layer is present. This is a gram positive bacteria. This is the peptidoglycan layers. This is the lipoproteins and cytoplasmic membranes. This is the gram negative bacteria. Gram negative bacteria is the only thin amount of peptidoglycan layer is present and outer membrane is a proteins is present. Then cell membrane. The site of biosynthesis of DNA, cell wall polymers and membrane lipids, selective permeability membrane is used to transport the solute into the cells. This is the important function for the cell membrane. Next, bacteria use the flagella and pili for the movement for the interaction with the environment. Pili is a common pili. This is the only hair-like structure is present in the outer cell membrane. Sex pili is for the conjugation for the reproduction. This is the flagella. Some bacterial species are mobile because of the presence of the flagella. Flagella consists of number of proteins and including a flagellate compound. The plasmids. Plasmids are small circular DNA. This is the extra chromosomal DNA. Then double stranded DNA also present in the bacteria. Nucleus. The lacking nuclear membranes only nuclear regions only present in the bacterial cells. The nucleolus is absent in the bacteria. Thank you.